Hi everyone, welcome to my channel. So this is part 4 and the last part for the chapter on infectious diseases in humans. Okay, and this is also the part that is the heaviest I feel because a lot of application questions can come from here. For the past few videos that I have made, the questions are pretty straightforward. But for this part, right, I would believe that actually there will be more application questions. So let's talk about vaccines and antibiotics. All right. Now let's talk about the syllabus first. So you need to state that vaccines contain an agent, okay, an agent that resembles a pathogen, and vaccines, right, prevent infectious diseases by stimulating white blood cells to quickly produce antibodies when the pathogen invades into the body. Okay, so generally, that's what vaccines do. On the other hand, you also have to learn antibiotics. So please be careful between vaccines and antibiotics because they're not the same, right? So you have to explain the antibiotics, right? Target bacteria by preventing the bacteria to grow. Okay, so I'm just mentioning this briefly, which I'll elaborate it in the next few slides. The last one, right, is something that is pretty real. Okay, it's actually very real. So you must learn how to discuss. So note the command word. Okay, discuss. Discuss is something about real life. So you must discuss, right? Why is there misuse and overuse of antibiotics, and how this misuse and overuse, right, will accelerate the emergence of antibiotic resistant bacteria. So this is something that is real, which I mentioned just now. Okay, and yeah, you can. It's probably something that I believe everybody should know also. So let's start with vaccines first. Okay, so normally, right, when a pathogen invade the body, our white blood cells, right, will be automatically stimulated. We will start to produce antibodies. Okay, and these antibodies will kill the pathogens. This is quite standard for all diseases. However, right, it might take some time. Okay, and by the time, right, it happens, when they start to produce the antibodies, it will be too late. Okay, and the person will be infected. So what vaccine does, right, is that vaccine contains an agent, excuse me, that resembles a pathogen. Okay, we use the word agent, but basically a substance. Okay, this substance, right, resembles a pathogen in many ways, three ways. Okay, the agent may consist of a weakened bacteria or virus. Excuse me. It may contain a dead bacteria or virus. Okay, it can be weakened or it can be dead, or it can be just a component of a pathogen. Okay, it doesn't have to be the whole virus or bacteria. It can be just one small part, but that part has to be important, of course. Okay, I've given you one example over here, and I believe if you remember your COVID lessons, you have probably heard of spike proteins. Okay, so what vaccine does is that it contains agents, and agents comes in three different ways. Either they are weakened bacteria or virus, either they are dead bacteria or virus, or they are just components of a bacteria and virus. Right? So when you introduce a vaccine, right, into an individual, it will stimulate the immune system to produce antibodies quickly. Okay, so in the process, right, it prevents the infectious disease from taking place. So it's like a preempt. Okay, vaccines can be taken orally or through injection. Most of you will know about the injection because of our experience with COVID, but there are also vaccines that can be taken orally. So let's move on to the more technical part about how vaccines work. Okay, now if you remember, right, there is a term known as antigens. Okay, but this antigen is not from the red blood cell in chapter 6. This antigen is found actually on the pathogen. So pathogens, right, on their surface, they also have antigens on them. Okay, so when a pathogen enters the bloodstream, right, the antigens, right, will stimulate the white blood cells to produce antibodies. Okay, so do not get confused between antigen and antibody. All right, I know it's confusing, but yeah, if you just keep on working on these two terms, you will not get confused. So the antigens on the pathogens, right, when they enter the bloodstream, the white blood cells will recognize the antigens, and the white blood cells will start to produce antibodies. Okay, these antibodies, right, will bind to the antigen, and after when it binds to the antigen, right, in the process it will destroy the pathogens. It will rupture the cell, or it will inhibit metabolism of the cell, so on and so forth. Okay, so again, right, a little bit of a revision from chapter 6 is the same phrase. The antibodies will bind to the antigen. Okay, not the other way around. Antibodies are proteins that are specific in their action. So if an antibody recognizes a particular antigen, right, they will just target that particular antigen and they will not target any other antigens. Okay, so they somewhat work like enzymes, but they are not enzymes. Okay, so they are specific in their actions. So if we introduce a weakened, a dead, or component of a pathogen through a vaccine, right, we can sort of like kickstart this antibodies production. Okay, so if we introduce them through vaccines, the white blood cells, right, will be stimulated to produce antibodies quickly. So before the person get the full contact of the disease, right, the antibodies are already in the body to counter the effect of the antigens of a real pathogen, or when the real pathogen, not real, or when the pathogen enters the bloodstream. Okay, then this white blood cells, right, when they already learn how to make the antibodies, okay, I'm just trying to explain to you in layman's term. When they already know how to make the antibodies, right, they can somehow uh, retain that memory, okay, and then it depends on the type of white blood cells, but they have a duration, and depending, some of them will stay in the body for very long. Some of them will not stay for very long, but generally they will learn how to make the antibodies. Okay, so if the same pathogen appears again, right, these white blood cells will stimulate and produce antibodies quickly. So it will be done pretty fast. And therefore, before the infectious disease takes shape, right, the person, the antibodies in the person, in the individual, will start to destroy all the pathogens and the person will not fall sick. So over here, right, this is a pictorial form that is taken from the textbook. Okay, for those who are more aesthetically inclined, because you like to study through diagrams, you can have a look. But generally, it is how I try to explain it just now in the previous two slides. Okay, so you can take your time to slowly read through. It is the it is the same. So let's move on to antibiotics. All right. Now antibiotics, right, targets bacteria. So they are drugs, right, that treat bacterial infections. Okay, they are not effective against viral infections. All right, so if you have a viral infection, for example, a flu, and when you take antibiotics, it is not to counter the flu virus, it is actually to treat the bacterial infection in the throat. Okay, for example, like sore throat. Antibiotics, right, are made from microorganisms. Okay, in this diagram, in this slide, I have shown you a fungi, and this fungi is known as penicillium. And of course, this is the one that gives you the antibiotic known as penicillin. So there are many types of fungi that research is trying to extract antibiotics from, and this is one of them. Okay, so generally antibiotics are from microorganisms, and because of technology, right, sometimes we can chemically modify the antibiotics to make them more effective in recent years. So how do antibiotics work? In a nutshell, right, antibiotics will kill or prevent the bacteria from growing. Okay, but how do they do that? There are four ways that they can do. So number one, they can prevent the synthesis of the bacterial cell wall. So if the bacteria cannot make the cell wall, right, when water enters the cell through osmosis, they will ultimately rupture. Okay. Number two, they can inhibit cell membrane function. So a bacteria is a cell. A cell has a cell membrane. The cell membrane will control substances moving in and out of the cell. If the antibiotic prevents the cell membrane from functioning, the cell will ultimately die, all right, because the cell membrane cannot function. In a bacteria, there are ribosomes, and if you remember your chapter 1, ribosomes produce proteins. There are many types of proteins, and if the antibiotic, right, prevents protein from being made by the ribosomes, ultimately the bacteria will also die. Okay, because there are many functions of proteins. In this case, I'm just talking about growth and metabolism. And number 4, I can also make the antibiotic, right, can also prevent the enzyme in the cytoplasm from working. Okay, somewhat similar to the previous point, but generally this is talking about enzyme, and enzyme speeds up reaction, right? So the chemical reactions in the cell, in the bacterial cell, will not be able to be performed optimally, and of course, in the end, the bacteria will die. Okay, so these are 4 ways in which how antibiotics kill or prevent the bacteria from growing. And these 4 ways are important now, of course. All right, you must remember all 4 because I can ask a 4-mark question, right? And then yeah, there you have it. All four points are there. Okay. So let's move on to the last part, which is the one that you have to learn how to discuss. When we use the word discuss, it is actually something of a real life issue, and antibiotic resistance is on the rise, and it is actually pretty um, serious, okay, right now. So how does antibiotic resistance in bacteria work? Okay, so generally, right, bacteria can grow resistance against specific types of antibiotics. Okay, so let's talk about a population of bacteria. Okay, a few billion bacteria in the population, right? Now, some bacteria, right, are generally more sensitive to antibiotics, and some bacteria, right, are less sensitive to antibiotics. Okay, so not all bacteria are made the same. Some are stronger, some are weaker. Okay, so we use the word more sensitive to antibiotics and less sensitive to antibiotics. All right, those that are more sensitive, right, will of course be killed because they are being triggered, right? The antibiotic will kill them. And for those who are less sensitive, right, they might not be killed. They might survive. And bacteria, right, they can reproduce pretty fast. Okay, they will grow and they will grow and they will grow. And those bacteria, right, that are not killed by the antibiotics, right, they will continue to grow and reproduce. Through time, right, the whole population, right, will be very less sensitive. That means they will become resistant. So if this antibiotic is being applied to this group of bacteria, the bacteria will not die anymore. And this is known as antibiotic resistance. So to destroy this new strain of bacteria, right, we have to invest in more money and more time, okay, to do research and development to discover new types of antibiotics. Okay, but in reality, right, it costs a lot. A lot of time is needed. Uh, Through politics and all. And the last one, right, is that our biodiversity is decreasing, which means that the discovery of new microorganisms will also um, not be found readily. Okay, so this is something that is actually pretty real. So to slow down antibiotics resistance, right, there are 3 ways that we can do it. Okay, number 1, do not misuse or overuse antibiotics. Number 2, when the doctor prescribe a dosage, you have to complete the dosage. In current years, it is 7 days, right? 10 years ago, it's only 5 days. And thirdly, to use antibiotics only when necessary. Okay, do not keep your antibiotics for a rainy day. You have to complete them. And yeah, so when you fall sick, right, the doctors will only prescribe you antibiotics if they think that it is necessary. This is the pictorial form for those of you who want to have a more visual feel for this. Okay, I'm not going to elaborate. You can pause the video and you can refer to it. It is also from the textbook. All right, so this is the end of part 4, and this is the end of the whole chapter. All right, so please study them. Okay, it is actually quite a lot, and try to remember as many concepts as possible. Okay, thank you. Bye bye.